My name is Menghang Xia, and I'm a group leader for the TOX21 program. The TOX21 team at the NCATS is made up of 3 groups. The first is System Toxicology Group for assay development and validation. We are working very closely with our robotic team for the compound screening. The second is Genomics Toxicology Group for the follow-up study. The third is the Informatics Group for the data analysis. In this video, we will give you a virtual tour of our labs. Now I will show you some equipment which we used for the assay development, assay optimization, assay validation, and compound screening. So first I will show you the cell culture room. So this is the cell culture room. So we have 8 cell culture incubators and 4 hoods. For the cell-based assay, we use a 5-layer flask to culture our cells because we need a lot of cells for our compound screening. Use of the 5-layer flask save us a lot of space for cell culture in the cell incubators. So when we are running the assay development, so we use the 1536 well plates. To add the cell into the assay plates, we use 2 types of the dispensers. The first one is a Bioraptor. So we commonly add 3 to 5 microliter of the cells into the assay plates. And we also can add 0.2 to 10 microliter of up to 4 different reagents simultaneously into this 1536 well plate. The other type of the dispenser is the multi-convey dispenser. So this is a high-speed dispenser, can dispense 1 1536 well plate within 20 seconds. So recently we got this new dispenser from the LGR company. This dispenser actually is much easier to use and also more accurate for the reagents volume. After cell will add into the assay plates, then we will add the chemical compounds into these assay plates by using this pin tool station. This pin tool can transfer 23 nanoliter of the compound solution from the compound plates to the assay plates. After we add the chemical compounds into the assay plates, and then we will incubate the assay plates for additional hours, depending on the type of the assays. And then we will add the detection mixture into the assay plates. And then we will read these assay plates under the plate readers. In our lab, we have 3 types of plate readers. The first type is the whole plate reader, such as the Velux plate reader, emission plate reader, and Felistar. The second type is the high content imaging reader, such as the Oplada imaging reader. So this type of reader, you can visualize and quantify many intracellular features on large scale. So we use this high content imaging reader to measure the neuroid outgrowths showing this screen. The third type of the plate readers is the kinetic reader, such as the FDSS plate readers. This type of reader can measure real-time kinetic reaction in the cells by measuring the fluorescence intensity in each well of the plate. So showing here on the screen is the calcium influx assays. To speed up our screening process, we use this robot system to run all the TOX21 screening. So in this robot system, there are 4 components. The first one is the assay plates incubators and compound storage. So we store the TOX21 library up to 4 months when we run the screening. So this library
containing about 10,000 chemical compounds, including industry chemical compounds, pesticides, food additive, and clinical approved drugs. This library is provided by EPA, NTP, and NCATS. The second component of this robot is a liquid handling system. In this robot, we have a Pinto station, and we also have a bioreactor dispenser, which I previously showed you before. So this liquid handling systems can transport nanoliter to microliter to the assay plates. The third component is the plate detection system. So in this robot, we have Viewlux plate reader, we have FDS kinetic reader, and we have Operada imaging reader, and we have the emission plate readers. The fourth component is the robotic arm, the yellow line here, and also the software controlling system to connect all these components together to finish running the screenings. So after the compound screening, Brady's group will process all the data analysis. So that's all the equipment I showed you previously for our TOX21 program. So if you get a chance in the future, you're welcome to visit our labs in person. Thank you.
Hello, I'm David Gerhold. I'm part of the TOX21 consortium here at NCATS, and I lead the genomic toxicology laboratory. I want to show you around the laboratory today and show you the equipment that we use, and tell you a little bit about how we collaborate both within the TOX21 program and with other folks at NCATS. We basically focus on two things in genomic toxicology. One is to develop new and improved cell culture models to represent normal tissues. And the second one is to develop new methods for doing high-throughput genomic analyses. And that also includes doing informatic analyses. And so we try to improve informatic methods as well. And we do a lot of that in collaboration with the informatics group, including Ray Lee. A lot of our cell culture studies are follow-ups on Monghong Sha's and Ray Lee's high-throughput screening results. And so we often take compounds that come from those results and we further characterize them. So here we have our cell culture heads. We have incubators. We have 3 incubators here. These 2 are standard. This one is dedicated for this IncuSight Zoom instrument. This is basically a glorified microscope which resides inside the incubator, and we can program it to scan up to 6 microplates at defined intervals. And so we will often do cytotoxicity assays where we monitor the growth and sometimes death of cells by scanning all the wells and microplates, usually twice per day. So that's the IncuSight Zoom. This is the external memory. And then results are first stored here, and then they're sent through a link to a remote server within NCATS. Cell culture is highly dependent, of course, on our microscopes. We have here a Revolve microscope, which is so called because it flips vertically like this, so we can look at flasks or slides from the top, or we can look at plates or flasks from the bottom as well. It also has fluorescence capabilities, so we can do fluorescence microscopy, which is important in our knockout and our CRISPR engineering studies. Here we have 2 biomech robots. The NX robot we basically use to transfer liquids between 96 and 384 well plates. We do most of our high-throughput genomics in 384 well format. And so the methods development and actually running methods, we do most of that on the Biomech FX robot here. This one has the ability to transfer things in 96 or 384 well format through this head in the back here. And it also has a set of 8 pipetters which can operate in concert or individually for more detailed pipetting procedures. It also can move the plates around, and we have a vacuum manifold attached so that we can do high throughput, relatively high throughput DNA or RNA isolation. We have a series of other robots or pipetters, really. I think of them as high throughput pipetters. We have 2 of these Integra instruments. 2 because this one is dedicated to isolate it so that we do not expose it to PCR products, which could contaminate our genomics procedures. So this one is no PCR products, and the other one is everything else. It's a simple pipetter, really, and we use this for lots of things where we're basically transferring 384 samples from one plate to another. The other kind of pipetting that we do a lot in our genomics studies is to add, for example, a little bit of enzyme to each of 384 wells. And we have actually 3 instruments that we use to do that. One is this, the newest one is this Funai instrument. And this is basically a inkjet printer head in a tiny little robot. And so it doesn't take up too much space. It's still rather new and a bit awkward to program, but this one works very nicely and precisely. We also have this older Mantis instrument, which has these interchangeable heads, which pipette a defined volume, and they pipette very quickly into a microplate, which sits on the deck here. And this is the arm that moves the pipetter and moves these little cartridges around, which will pipette, for example, 0.5 microliters into each well. The third instrument we don't have in my lab, but we share. That's the Bioraptor 2, and everybody in NCATS uses these Bioraptors. So I think we have, last count, we have about a dozen. This is a Bioplex 3D Luminex instrument, which we use to do ELISA assays. We typically use this to monitor 42 cytokines in the medium of our 96 well samples. It's a bead counter, basically. And so the assays are immobilized on the surface of different colored beads, and it rapidly counts the beads and quantifies the amount of each cytokine on each bead. This is the Envision. This is a plate reader, and we have probably lots of these at NCATS as well. We use this to quantitate in 96 or 384 well format for quantitating fluorescence or luminescence. This is called a Quant Studio. This is a quantitative PCR instrument. This one is relatively high throughput. We've had this for a couple of years. We have a bank of regular PCR instruments. The method we're developing now, called Apples for screening by gene expression, requires us to do lots of annealing steps into many plates at once, so we can apply it for high throughput screening. So this is basically a programmable heater. And so we'll put a whole stack of plates in there and run it through a defined temperature program. Here we have a caliper instrument. This does capillary electrophoresis. We'll put 96 or 384 samples in the instrument. It will sip them one by one, and it will size the DNA or RNA fragments that are in each sample. So this has been indispensable for methods. And then finally, all of our genomics methods end with next-gen sequencing. And so we have a MiSeq instrument, and we have a NextSeq instrument. This is a NextSeq. They look about the same. And so when we do methods development, we do our own next-gen sequencing, and we can turn things around quickly. When we do very high throughput, like screening, or we're generating lots of data, we will actually send those samples out to a sequencing core facility. This is an echo. This is an acoustic dispenser. We use this when we want to dispense small amounts of liquid, usually toxicants or drugs, into microplates. And it works based on acoustic energy. And so it's not as fast as the pin tool that Honghan will show you, but it's very precise, and we like it a lot. Also, we're getting, like everybody else, we're getting into doing cell engineering. We're doing some CRISPR to engineer our neurons. And here we have a preparative flow machine. So this is a FACS machine, a fluorescence activated cell sorter. It works on a preparative basis and it has a disposable chamber for doing the sorting. And so we'll use this to sort transfected from non-transfected cells, and that enriches the cells that have been engineered by CRISPR. And so this is another incident that we shared in the
Hello, my name is Ruili Huang. I'm the group leader of the Tox21 Informatics group. So we are responsible for processing all the QHTS screening data generated by the systems toxicology group, Mohan's group, and also genomics toxicology group, that is Dave's group. So within the informatics group, we developed a set of software tools to help with the data processing and data analysis. So I'm going to show you some of the software applications we have on my computer. So we start with, we have a set of Java clients. So this one is called the console client. So this one is used to load in the high throughput screening data that come off the robot. So here there's a data loading tab, and also we have a data normalization tab. So this is data correction. So you're seeing here is the plate images of the screening place data. So here it shows the raw data, and then the data after correction, and then those are the residuals. So you can see a big difference before and after data correction. So after that, we'll go ahead and pivot data into concentration response curves, and then run curve fitting. So we also have another software Java client called the plate client. So here you can get detailed views of the plate level data. And so here are some of the performance statistics, like signal to background ratio, covariant variance, and also Z factors. So you can go through the plate views. So after curve fitting, so we use another Java client called the QHTS client to see the curve fitting results and the concentration response curves, just showing some curves and compound structures. So after we finished with the data processing and got the concentration response curves, we'll put the data into the public domain for the entire scientific community to see. So here, this is we have this is called the Tox21 data browser. So this is the public data. So here you can see all the assays that we have and different cell types. So here is a list of the assays that we have data on. And then you can either go to here to download the data. So there's a set of zip files, or you can go to each individual assay. So here we also have assay descriptions, and then here you can download the data. So we also have, go back here, a Tox21 compound library page. So here we have all the compound structures in the Tox2110K library. And also, because all the Tox2110K compound library have gone through analytical QC to determine identity and purity of the compounds. And all the QC results are publicly available from this web page. So if you click on a compound, and then you can also see the QC grade and the actual spectra of the QC traces. So we also have this tool for structure activity relationship analysis. So the entire Tox2110K library have been clustered by structure similarity. And then, so here, this heat map, every little hexagon here is a cluster of compounds with similar structures. And then the color of the clusters indicates the number of active compounds in each cluster. So if you can right click on the cluster and then look at the actual AC50s, AC50 profile, for example, for this cluster of compounds across all the Tox21 assays. And you can also look at the structures of the compound in each cluster. So for gene expression data analysis, we have developed this called a point of departure website. So this determines the point of departure for dose response gene expression data. So the point departure is the concentration at which a chemical induces a significant response, gene expression response. So here, if you click on each row, then you can see the concentration, gene expression concentration response curve of the compound. And then here we have, so those columns, POD fit is actually the point of departure for the for that chemical. And we also assign curve classes to the concentration response curves. So this page also provides download, so you can export the data. Just enter your email address here and then start download. So help with the data analysis, we also developed the this is called the BioPlanet. So this is a database of all the human pathways. So currently the database has around 1,700 manually curated human pathways. So you can browse the pathway. So under each pathway, we provide the genes and disease relevance, also assay availability for a particular pathway. And here you can search by, for example, a gene ID, and then you get all the pathways for in this case related to p53 signaling. And then you can look at the pathway map. And then all the genes in this pathway. So if we search for toxicity, then you get all the toxicity related pathways. And you can also project this onto this. So this is why this is called the BioPlanet, because we have this globe that shows the universe of all pathways. And then each dot on this globe is a pathway. So those are all the toxicity related pathways on the BioPlanet. So those are all the software tools that we have to help with Tox21 data processing and analysis. I hope you enjoyed the tour, and thank you.